start.
Yes.
Helen, tell a little bit about you. What you did before you were in the Santos. So she lives in the city, formerly from Pelé, the famous soccer player. Yeah,
Yeah.
He's passed away last year. Santos City. Yeah.
Well, it's a pleasure to be here with you. Thank you, Marcelo, for the invitation. And my name is Helen. I am a biologist. I work especially with ecophysiology and ecotoxicology of aquatic organisms. Now I am working in a university in Santos City, located in São Paulo State in Brazil. I am a professor, work with research and teach also in the same research area that I made my PhD and my postdoc. So the work that I will present for you today is a work that I developed during my postdoc in Manaus City, so in Amazon, and together with a professor from the UBC, University of British Columbia in Canada. So Christopher Michael Wood, I don't know if you know him, he's a very good research in this area of physiology. So these experiments that I will present for you today, I made there, so in UBC in Canada. I also was in the island, I will show you. And after my postdoc, I come back to Brazil and start working here in Santos City. Okay, so I will share my screen with you to start my presentation. And if you have any doubts, I don't know how is the dynamics here, if you can ask something in the middle of the presentation, or we do on the final of the presentation, Marcelo. Okay, you can ask if you want to, okay. I will share my screen now. Let's see if I can do this. I never used this app. Okay, can you see?
Yes.
Okay.
Yeah, yes.
Okay, so this work, I performed it together with the UBC University, but the practical part was performed in the Banfield Marine Sciences Center, located in Vancouver Island, close to Vancouver City. And there I performed this experiment. So the article is already published. So we published this in the final of the last year. So it's on the Journal of Experimental Biology. And the work was financiation for several organs, like the federal one of the Amazon, CNPq, CAPS, Science Without Borders, that I have my fellowship by it. And from the ADAPTA, there is a big project that we have there in Manaus City. So the title of the work is Emersion and Recovery, Alter Oxygen Consumption, Ammonia and Urea Excretion, and Oxidative Stress Parameters, but not diffusive water exchange or transepithelial potential in the green crab, Carcinus maenas. Well, why do we choose to study this little guy here? Because this crab is an invasive species, so he has the capacity of live in different types of environments. So it has a lot of success in staying in these environments. So the crab lives in the intertidal zone, so that area that stay exposed to air, some emerged some periods of the tide, and submerged in other periods of the tide. So these dynamics makes these organisms to have some biochemical, physiological adaptations to deal with it. So that's we would like to study and understand how they can stay in these environments in several countries, several parts of the world, because it's a exotic species in a lot of places. So these organisms that live in this intertidal zone, they are adapted to extremely dynamic conditions. So they have not only biochemical and physiological responses, but also behavioral responses to physical chemical characteristics like temperature, salinity, dissolved oxygen during these periods of immersion. And these periods of immersion is one of the most stress-inducing situations. Why? Because the organisms stay vulnerable to the desiccation stress, which is associated with their exposure. So during these periods, these organisms can decrease the efficiency of respiration and lead to eventual collapse of the gas exchange and aerobic metabolism. So these features are very important for us to start to make our questions and postulate our hypotheses, to study these organisms and understand how they can deal with this immersion stress. Okay. So the objectives, the main purposes of this work was to evaluate whether 14 hours of air immersion followed by 14 hours of re-immersion recovery would influence the rates of oxygen consumption, ammonia excretion, urea excretion, some oxidative parameters indices, and water conservation and gill permeability in these green crabs facing this immersion period. So we choose 14 hours because we made some research and we know that these organisms can stay immersed in air during this period. So they actually deal with this in natural environment. So we would like to reply this in the lab to analyze these parameters. So we only work with male green crabs once the females have some difference in the behavior, and they can actually, we also did not choose females because it was in the period that we could not collect the females from the environment. So this is the Bamfield's Research Century. So we have a lot of facilities to work in it, and we have the support of fishermen and technicians and everything. So we collected the green crabs from here and took them to the labs. So they stay armazened here to acclimatization. And we performed 4 different experimental setups, because one of it we analyzed different parameters. So first of it, we made an experiment to measure the MO2, that is the metabolic rate or oxygen consumption, is the same thing. And also we collected tissues for analysis of oxidative stress parameters. After, we performed another experiment to measure the ammonia and urea excretion that is related with the metabolism of this organism. The transepithelial potential, so how permeable these tissues was, especially the gills. We measured this to know, actually is an indirect way for us to see how the gills was permeable or not regarding this air exposure and recovery. And we also made another experimental setup to check the water metabolism. What is this? We can see how much water they are losing when they are air exposed. So we use radioactive water, is tritiated water. So it's a marked water, then we can see where this water is, you know, if you are inside the organisms or outside the organisms. So the first setup, so when I get there, we have a really good structure, but they are some they like to build their stuff. So I have to build my camera, my respirometer. So here is a picture of one of it. So I made it using these pots and some syringes and some equipment here to collect the water, to measure or the air, depending on the treatment. And I can use these to measure the oxygen after the experiments. So I use the crabs to expose to water, air, or recovery in these glass pots and containers, actually, it's a better word, and these glass containers. So they were individually distributed because we have to measure the metabolic rate of each individual. So they were individually put inside these containers. And in water, they stay 14 hours submerged. Then they stay more 14 hours immersed. And after that, they were put on the recovery. After they stayed 14 hours in air, they go back to water, re-immersed again for 14 hours. Okay, so they stay 28 hours after these two treatments for the recovery. So 14 hours in the water, more 14 hours in, 14 hours in the air, then more 14 hours in the water. And this was my recovery treatment. So was a lot of work because was a long time doing the experiments, but with really nice results. Well, the water was collected from by this syringe here. So I pumped the water from inside the container. And on the initial and the final, so I here, PO to AI is the initial oxygen concentration, and PO to F is the final concentration. So I collect on the beginning and on the end of this 14 water treatment exposure. So I have just two measurements, but for the air, I made the collection more times. So I do at 30 minutes between these 14 hours in air. Okay, so I have a better accuracy to analyze the oxygen consumption of these crabs when air exposed. So this is the first experimental setup where I used this to calculate the metabolic rate. So after the 14 hours in air, water, and recovery, the organisms were sacrificed to collect the tissues. But before, I will just show you the equipment that I use there to analyze the oxygen in air and in water. So we use this equipment to make this analysis. We have a formula to do this calculation. So the metabolic rate is the difference between the initial and final oxygen concentration in the containers times the coefficient of oxygen diffusion in water times the volume of the camera divided by the mass of the crabs times the period that they were in this experiment. So the crabs, after these 14 hours, the crabs were sacrificed, and we removed the gills. So I divided the gills in two parts, the anterior and the posterior. The anterior was the 50 arc, and the posterior the 80 arc, because we know that in crustaceans, these different parts of the gills are related with different physiological processes. So the anterior are related with the osmoregulation, the posterior with this more biochemical process, not just osmoregulation. Also, I collected the petopancreas and the muscles to analyze T-bars. So T-bars, we measure T-bars to know the lipid damage, so how much these lipids molecules was oxidized by free radicals. The same we can see in protein, and we also analyze the catalase, that is a antioxidant enzyme that act neutralizing these free radicals. Well, for the second experimental setup that I performed to analyze the ammonia and urea excretion, actually I did the same from the first one. Okay, the same pots, the same containers, the same volume of water, everything. But instead of collecting water for oxygen, I collect water for measuring ammonia and urea. Okay, so it's the same but different thing, not for oxygen, but for ammonia and urea excretion analysis. I try to bring for you some videos and photos from the 30 and 40 experimental setup, but I couldn't find it. So I couldn't find the videos, and it's hard to explain these experimental setup because it's really full of details. But what you do is to measure this transepithelial potassium, we just measure the potential difference between inside the animal and outside the animal. So with this potential difference of electrical potential, we see if the gills are more or less permeable. Okay, so if somebody would like to know more details about this methodology, we can talk later, and okay, I can explain better. But basically is that we make a hole in the crab's carcass with them alive, of course, they cannot die, and we make this hole and cover this hole with some glue that is not toxic. And when we need to measure this different electrical potential, we just open this hole and put the electrode inside and outside, and the equipment show us this difference of electrical potential. And we might have some calculations too, but I didn't bring here. If anybody needs to know more about this, I can talk later about it. And for the 40 experimental setup for water measurements, so we just the same as the first and second one, we just give the crabs a bath of tritiated water, radioactive water, and then after that they incorporated this water in their body, we just use them to make the experiment, so their exposure, the immersion, and the recovery, and measure how much of this tritiated water was outside of the crabs. We use a liquid scintillation counter to analyze this tritiated water. Well, what we got after our experiments. So what we see was the crabs in air. So during the air exposure, they keep their metabolic rate low. They don't use a lot of energy, let's say, like this. They don't use a lot of energy inside, just like they were in recovery. So in the air, they just slow down their metabolism and stay quiet to keep energy, and then though increases their metabolic rate. But when the crabs, after air exposure, was put back in the water, so the recovery treatment, they increased significantly their oxygen consumption. So this shows us that these organisms, after the air exposure was trying to recover their normal metabolism. So that's why they increased so much the metabolic rate. So no difference were found between water and air crabs, but when they were put back in the water after air, they increased a lot, significantly increased their metabolic rate. Well, and what happened with the ammonia and urea excretion? So ammonia and urea are a product of the metabolism. So when we have increases in metabolism, we can increase this excretion of ammonia and urea because it's a product of our biochemical processes. So we see here that crabs during the air, they almost don't excrete ammonia and urea. So this is in accordance with our MO2 response, so the metabolic rate. So as they were quiet, let's say like this, during the air, they don't spend a lot of energy, so they are not excreting ammonia and urea. But when they were back in the water after exposure, they increased a lot the excretion of ammonia and urea, so significantly increased the excretion during the recovery, and during the air, they significantly decreased the excretion. All right, so this is what indicates to us that they have this increased metabolism that we saw during the MO2 analysis, the metabolic rate. We see that they really have an increase of these parameters excretion. What we see for oxidative stress, so we expected that we did not have a lot of oxidative stress during the air exposure. Once they were quiet, they were not metabolizing too much, they were not spending a lot of energy, so we don't have a lot of products of the metabolism. But what we saw was that so this is anterior gills, this is the posterior gills, and this is the T-bar, so damage in lipids and damage in proteins, okay? So lipid damage we saw in anterior gills when crabs were air exposed, just for anterior gills, not for posterior gills. And we didn't see any changes for carbonyl proteins, so no damage in proteins were observed in anterior or posterior gills, just T-bars in anterior gills when crabs were air exposed. Here we have for anterior and posterior gills also, so anterior and posterior, the same, but here we have catalase. I have, sorry, can you hear me? Because I think, wait, okay. My computer is not working really. It's way. Okay. So here we have for hepatopancreas, and the response for oxidative stress in hepatopancreas was more interesting, I think, because we didn't have any damage in lipids, but the catalase decreased during the recovery, and we argue that the catalase increases could protect the hepatopancreas from the lipid damage that we didn't see here in recovery period. Okay, so we did the same parameters in the muscle, and we didn't have any difference in the muscle. Well, we expected that the gills from crabs during the air exposure and during the recovery had some difference in electrical potential, but we didn't see it. So how we interpreted this step, that is the transepithelial potential. So when it's close to zero, more tight are the gills or the tissue, so less permeable are the tissues, and how far from zero to negative values, when close to zero, more permeable are the tissues, far from zero, more impermeable are the gills. So what we see here, we didn't see any significant differences between the transepithelial potential, okay? So what we see is the crab could keep this electrical difference potential between inside and outside, that this could be related with the capacity of these organisms to keep the absorption and excretion of some important ions like sodium chloride, but we didn't measure this in this experiment, so we just speculate this. For water exchangeable pool, we also didn't see any significant difference. So the crab was able to keep their pool of water. So even if they were air exposed, they did not lose water to the environment. They could keep the water inside. So the crab did not desiccate it. So this shows us that the immersion is not a problem for him, because they can keep their water pools inside constant, so they don't desiccate. So our conclusions was that this huge ability that these species have to survive prolonged periods to air is probably an important contributor to its success, of course, for being an exotic invader. And we demonstrated in the present study that these species can tolerate 14 hours of immersion with a minimum disturbance in permeability and water metabolism, and can maintain their metabolic rate at a low resting level. So this is really important. They keep their metabolic rate in a low resting level, so they can not suffer from these metabolic rate increases. But we didn't see changes in some parameters, but these metabolic costs increased this epoch, that is post-immersion oxygen consumption. So we see these epoch responses during the re-immersion recovery. So the huge problem for crabs is, okay, they can deal with the immersion, but when they were re-emerged, some metabolic expenditures occur. So this was seen in ammonia excretion and urea excretion that greatly elevated during the recovery. So the metabolic costs of the re-immersion was huge. And the oxidative stress was manifest as a lipid damage just in the anterior gills, which is specialized for respiratory gas exchange. And we saw that the catalase activity was important to prevent the T-bars activity or the lipid damage actually in the pathopancreas. And this damage was not seen in the posterior gills that are specialized for osmoregulation. So the osmoregulation part, let's say like this, of the gills didn't suffer from oxidative stress. Just this is say for us some specialization of these gills of these species, because the osmoregulation is really important for the surviving of these animals. So if they have damages on these membranes, that where we have most part of the membrane transporters, to keep the osmotic regulation would be a problem. So the damage will occur just on the anterior gills that protect, let's say like this, the species from this type of damage that they suffer every day. They every day are exposed to low tide and high tide, so they are emerging and re-emerging all the time. So that's it. I hope you enjoy it, and you have any doubt, I am here. Thank you.
Hello, Ellen. Thank you for your talk.
You're
Thank
here.
you.
I cannot see you. Wait a minute. Let me open again
It's
here.
me, it's Marcelo. Can you hear me?
Okay. Yes.
Yeah, I am my camera, something wrong. Thank you for your talk. It's very interesting. I have a question. Did you measure other antioxidants in these tissues?
No, Marcelo, we didn't do it because I had just 1 month in the station. So I have a really short time to do it, all these experiments, yes. And also, we didn't have a lot of structure because we have everything that we forgot, we have to bring from the city. So we didn't plan to do other things. We just planned to do these parameters.
Okay. But if you left some material there, maybe other people could measure, because that would complete your conclusion.
Yeah. It would be very nice to do another enzymes and osmoregulation enzymes too, but I didn't have time. Actually, I have less than 1 month. Was 20 days. Was insane.
Wow, but that's a lot of work for 20 days. Amazing. Wow. Oh, congratulations. Yeah. So please send to us the paper you publish in JAB. Yeah.
Yes.
Yes. Send the paper in the group.
In the group, yes.
Yeah. That'll be nice.
Mm-hmm.
Okay. People ask questions.
Helen, it's me.
Hi, Professor. Hi.
From Greece. Okay, very nice work. Very nice talk. Congratulations.
Thank
Really, it's a very interesting topic. And as far as I know, this species is very well accessibly studied crop. Okay. Regarding the ability to adapt such so different changes, environmental changes. Really, I was surprised seeing that you used Copenhagen radiometer. I was using this device many years ago.
Really?
Yeah, intracellular and extracellular pH. And let's say the question, could you calibrate this microelectrode with corresponding temperature? Have you this ability to do that?
Calibrate? Sorry, I didn't understand.
Yeah, when you measure oxygen consumption, okay, probably you determine oxygen consumption at the temperature to which you expose the animal, the species.
If I can, we consider this when we put on the formula. We use the oxygen coefficient, oxygen solubility coefficient. So it's a table that we can find in books of physiology. So I use on the formula the coefficient from the temperature of the experiment.
Okay, you use the formula from the tables.
Yes.
That's okay. Okay. And okay, when you expose the individuals there, what temperature you applied?
Yeah.
And
So
it was the same after recovery, re-immersion?
yeah. So what I did was to use a water bath. You know, a water bath. So I put
Okay.
yeah. So I use the water bath and keep measuring the temperature and keeping the temperature the same for the 3 treatments: water, air, and recovery.
Yeah. Do you use several treatments with temperatures, different temperatures, or one? 13?
No, it just was one temperature was I think 28 degrees, and I don't record it. We used the temperature that was in the experimental room, that was a climatized room.
Okay, only one.
Mm-hmm.
We did experiments with different temperatures.
No, I just fixed the temperature in 28, and all the experiment was performed in 28.
Because under natural conditions, this species faces entirely different temperatures. I mean, high
Yes.
temperature. I don't know what is the temperature, air temperature in this area
Mm-hmm.
where you collect species, individuals.
Yeah.
And
Yeah,
that
yeah.
would be nice if you could try this. I mean, high air temperature,
Mm-hmm.
and during the immersion, if the temperature is probably lower,
Yeah, you're
to
right.
see this difference.
This difference, yeah. Yeah,
Okay.
because actually we have a big difference between the temperature in air and water. Yeah, we didn't consider, actually we didn't want to use the temperature as a factor. We just fixed that temperature to check the immersion responses. So we didn't.
And yeah, according to your first data you show us, if I remember well, oxygen consumption was lower during air exposure?
Yes.
Because I couldn't see that. I mean, the bar shows a little bit higher than the control, or not?
No, for air, for MO2, that is the metabolic rate, the first
Okay.
graph.
Okay.
Yes, the first graph. So the metabolic rate was really low during their exposure.
And really, I couldn't relate it with high metabolic cost, since we have lower oxygen consumption.
Yeah,
Probably
but
you didn't determine an ATP, all this energetics.
No, we
To
just
see what happened.
speculated. Yeah.
To see whether we have a metabolic depression or not.
Yeah, we didn't measure it. It would really interesting to do that, but we didn't measure.
So the question is, how you come in such a
yeah.
suggestion.
But what we say is that during the re-immersion, that the metabolic rates increases a lot. So that's what we saw during the MO2 measurements. So the metabolic rate was really low during their exposure. And when they were put back in the water again, the metabolic rate increases a lot. So that's what we was trying to say is the metabolic costs for the air exposure and re-immersion is really strong.
During re-immersion.
The re-immersion, yeah,
But
the re-immersion.
During air
Air,
exposure.
water, no, just after air and water. So during the air, they are with behavior low metabolic costs.
Yeah, it is reasonable.
Yeah.
Okay.
Yeah.
And I couldn't really understand why did you determine lower catalase activity during re-immersion, since during lower activity or catalase activity.
Okay.
Catalase. Okay, sorry. And because as far as I know, ROS are produced during the immersion when we have higher oxygen consumption.
Could you repeat, please? I didn't understand.
Yeah. ROS are produced, according to
Okay.
several investigations,
Mhm.
during recovery
Okay.
from anaerobic to aerobic metabolism.
Okay.
And I would expect higher catalase activity in order to eliminate the ROS production.
I perfectly, yeah, that's what I thought about it was just during the air. So the catalase was working a lot to try to neutralize this oxygen reactive species. So for when during the recovery, we had a decrease of the catalase because they were all maybe consumed during the air. I don't know. That's what I tried to postulate it about this response of catalase and TBARS for hepatopancreas, because we only have responses of catalase for hepatopancreas.
Mm-hmm. And the last one question, sorry, so many questions.
No, that's okay.
But this is very interesting. Why you did examine the heart tissue? Heart tissue. Heart.
No, we use gills, hepatopancreas, and muscle. Why? That's a great question. No, I don't know.
It's the most aerobic tissue.
Yeah.
Many mitochondria. So it's a very good indicator of what is happening in the body.
Yeah.
I know it's my...
No, you're right. Yes.
Okay.
I could repeat the experiment. We could repeat it and do.
I don't say that. Okay, thanks a lot, really. And yeah, congratulations. I'm okay, Marcelo.
Oh, thank you for your questions. You make so many questions. So next time I have a thesis from my students, I will invite you.
Yeah.
Make them a lot of questions, you know.
That's good. Yeah.
I always get to pick up the most difficult referees for students.
Yeah.
No, you're the guy.
I hope that addressing many questions, probably I'm helping the students. Okay.
Yes. I'm just joking, but it's good to have someone asking a lot of questions.
Yeah.
Okay, people, more questions. It's open for questions. Let's take the opportunity. Don't be shy. I have one more question. What are you planning to do now? Now you're back in Santos.
Okay, regarding the research? For research?
Yeah, what are plans for research now?
Yeah, I'm working. Actually, I'm not working more with, I couldn't start working again with air exposure and recovery, everything. I am working more with ecotoxicology now. Not too much ecophysiology, but I would like to back to it. Actually, we are working together on that study. Marcelo, do you remember the girl that worked with you in... Do you remember that? She went to your lab last year.
Oh, yes.
Yeah, we need to finish that work. That is, corals are exposed. I had another student working now on the Sebimar. Do you know, remember Samuel? I don't think he's here. No, he's not here. He's on vacation, so completely off. But we made some collection on Sebimar, where we collect the corals from when they were air exposed and during high tide, low tide. We collected them, put them on the lab, and we made this simulation of the tide.
Oh, that's nice. You did that with Samuel.
Yeah, and my master's student.
Okay, yeah. So that will be very complementary to Marina's plan for PhD, because she wants to do this in nature, you know.
Nice.
Now we're hunting animals in the nature. Labs only for measurements, no more experiments in lab, at least for these big animals. Yeah.
Yeah, the idea is, our idea was to collect the corals that normally did not stay air exposed, because we have some corals that never are air exposed, and they are from the same species. And we will see in lab how they deal with their exposure if they are not used with it. So we will see if the responses, the corals that always stay air exposed will be the same from the corals that are never air exposed. So that's the idea.
Oh. Yeah, they might die soon. Yeah.
They did not die. We did the experiment. They survived.
Oh, that's
No,
good.
they did
That's
not
interesting.
die. Yeah. Now we're gonna see the response. So in this experiment, we will measure a lot of more enzymes and raws and everything. It could be more complete.
Oh, that's good. That's great. Thank you. Thank you. Okay. Does anyone have more questions? Please don't be shy. Marina, do you have questions?
Hey, Marina.
She's the one coordinating. See, this is our 7th seminar for our club, so it's working. Okay.
I was really happy with Marina's invitation, and I hope I could come back with more results of other experiments, and maybe my students from this one of corals could present for you too.
Okay. Yeah.
But Professor Basile will make a lot of questions for him, because he's a master student. We need that.
Are you going to continue this kind of experiments, co-physiology experiment?
I would like to continue, yes.
Yeah. Really, I could recommend you to use, since you have this condition over there, I mean, long periods with low tide, high tide, and the different temperatures when they are air exposed, immersion, re-immersion, etc.
Yeah.
You could simulate the crabs. You could make some simulations with using little sensors, robomuscles. You know, Marcelo, you know robomuscles? These are little sensors always recording the body temperature.
Oh, okay, little. So those are small bots. Yeah, to measure. Yes,
Yeah,
I know.
you can simulate it, and you can keep animals, individuals, out of the water for several hours. Okay, and then always record the temperature body recording. Isn't necessary to use many such sensors. That's a couple. Okay, and then when they are going back to the sea, to record the difference body temperature, how it changes. It's very nice to see what's happening with oxygen consumption, how they influence the recovery this species.
Nice.
That's
really nice idea. Thank you. That's a really good one. We'll take notes here, and maybe next experiment we can try do that.
It's very easy to find these little sensors.
Okay.
Really.
Okay. Is there anyone more with questions? So I think people are shy, or they have no questions. Okay, Helen, thank you very much. Thank you.
Thank you.
Sergey saying he asked a question in the chat. Sergey from Russia.
Yeah, I can read all the questions. Can you hear me? I think Oh,
Thank you very much. Maybe you have an explanation why there was lipid damage and not protein damage. He asked why lipid damage and not protein damage. Okay, that's a good question.
Can you hear me? Because I am not seeing anybody anymore.
Yeah,
okay.
Yeah,
so that's Sergey. It's a really good question, and also actually was one of the referees of the paper question. And what I speculated was that for maybe the lipid is more suitable for damage than protein, that it's more important for the cells. I don't know. That was we speculated, but I don't have sure. I don't know. Maybe if you have some idea. But we only saw damage in the lipids, not in the, maybe we have more time of damage. It could happen in proteins too, but don't know.
Does it have to do anything with lipid oxidation as a fuel for energy? I don't know, it just came to my mind. Like when they are stressed, this lipid oxidation as a stress factor is something like a switch, metabolic switch for energy consumption. That's why the first thing that came to my mind is like, okay, this is going to the stressor as a metabolic fuel than the protein carbonylation, which has to be stable. I mean, not any organism wants to spare its proteins for energy, or it's the last thing to be consumed.
That's what we speculated on the paper too. It's the proteins could be the last thing to be consumed. Yeah.
I'm the academic child of Professor Michailidis, so I'm starting to ask some questions also.
Okay.
Thank you very much. It was really interesting.
Thank you, and sorry about my English. My English is not really. I lost my words, and that's the problem. There's a long time that I don't do a seminar in English, so maybe sorry, but you could understand my whole thing.
It was great. It was great.
Okay.
Oh, no, your English is very good. That's no problem. Okay, people, more questions? Do you have more questions, Costa? More commentaries?
No. I'm fine, thank you.
Okay. So I think
Marina say something here. Marina said, I'm not sure. Yeah, she said that she found some difference in her antioxidant responses after 10 minutes of resubmersion. But that's a good point too, because we wait 14 hours of re-immersion. So maybe our responses start on the beginning of the period. This was a nice thing to do also. Do the collection of tissues along different periods of the re-immersion in water. So that's a good point, Marina.
Yeah, yeah. 14 hours, a lot of things may happen.
A lot of time. Yeah, a lot of things can happen. Yes.
Usually damage, it's nice to measure within 1-2 hours maximum to see things. Yeah. But maybe for your physiological measurements, it's not enough. So it's
that was another thing that referees asked us about it, is why we took so long to make the measurement. So why we keep them 14 hours recovering, but it's what we would like to simulate the tide. So that's why. But to see what's happening in all biochemical responses, we should start after 10 minutes, 30 minutes, 1 hour, and etc. But we didn't have time and crabs and hands. we could do
Yeah,
it.
yeah. It's we're always gambling with how many hours we're going to expose, because we don't know. We can't predict. Yeah, we don't have a crystal ball before the experiment. Yeah. Okay. Are there more questions? No, I think we people are quiet now. Okay, Ellen, thank you. Thank you very much.
Thank you.
It was very nice to hear this experience. You know, it's incredible that you did all this work in a few weeks.
20 days. Yeah.
Yes, and got this published in a very nice journal. And okay, thank you everyone. Can you guys open the camera so we can take a picture for everybody, if it's possible? Well, my camera is not working. Can people open the camera just to say hi?
Hi.
Green screen. Okay. Oh, hello everyone.
Hello.
Okay. Thank you, Ellen. So we're hunting for someone for the next seminar. Yeah. Maybe I invite Professor Costa if you have, or Professor Max. We'll see.
Okay.
Or Professor Marcelo Hermes.
Yeah, maybe. Yeah. But I want to hear the other ones. Yeah. The younger people. Yeah. Okay, people. Thank you very much. So we'll see you next time.
Thank you, Marcelo, for organizing these really nice seminars. Okay.
Thank you, everybody.
Bye.
Thank
It's
you.
a pleasure.
Bye, Helen.
It's a pleasure
Bye.
to organize these seminars. Yeah. It's a big pleasure. And thank you for the ones that participated.
Thank you all. Thank you, Helen.
YouTube in a few days. Okay. Bye-bye.
Bye.
Bye bye all.